Good afternoon. I'm Larry Biegelsen, the Medical Device Analyst at Wells Fargo. Welcome to the next call in our 2026 MedTech Innovation Spotlight Call series. I'm pleased to have the management team from Bausch Lomb. Speakers for this call will include Dr. Yehia Hashad, Executive Vice President of R&D and Chief Medical Officer, Dr. Mayssa Attar, Senior Vice President of Pharmaceuticals and Consumer R&D, Dr. Kelly Swain, Senior Vice President of Surgical R&D, and Dr. Kathleen McCabe, an ophthalmologist and cataract surgeon who's a strategic medical advisor to Bausch Lomb. Additional participants from the company include Brent Saunders, Chairman and CEO, Sam Eldessouky, Executive Vice President and CFO, and Andrew Stewart, Global Pharmaceuticals and International Consumer President. On today's call, we will focus on Bausch Lomb's exciting glaucoma pipeline, specifically BL1107, its alpha-2B agonist, and Elios, an excimer laser for glaucoma. We'll start with a presentation by the company that summarizes the overall market and opportunity, the products and their mechanism of action, and development updates. We'll then proceed to Q&A. And if anyone has a question they want me to ask on their behalf, please email it to me. So everyone on the line from Bausch Lomb, thank you for the opportunity to host this call. And at this time, I'd like to turn the call over to Dr. Hashad.
Thank you very much, Larry, for the opportunity and for hosting us. I actually will start by a very brief introduction, then I will hand over to my colleagues to give you the details about the projects. Can we go for the next slide, please? I think we covered who will be attending. I just remind everyone, obviously, end of last year at the Investor Day, we presented our pipeline. Earlier this year at JP Morgan Healthcare, we also presented the key milestones that we will have in 2026 for our robust portfolio of pipeline. I think today we are going to focus on the glaucoma assets, which are highlighted here in yellow. One is related to BL1107, the first glaucoma anti-protective, neuroprotective product, lowering intraocular pressure and addressing also functional outcome. And then Elios, the laser trabeculostomy, we will explain also, and its targeted approval also in the second half of this year. The rest of all other products are tracking well for this year towards achieving their corresponding timelines and milestones. Next slide, please. Before I turn it over to my colleagues, I just would like to give a very brief information on glaucoma. So what is glaucoma? What are the types of glaucoma? How we manage glaucoma, and also speaking about some numbers related to the prevalence and incidence. So glaucoma is defined as a chronic neurodegenerative condition. In fact, this even considered a new definition, because in the past, glaucoma was defined in relation to intraocular pressure. But what happened is that we found out that there is a lot of people that could be also having a normal intraocular pressure, but still have neurodegenerative condition that is very similar to what we see with the elevated intraocular pressure. So that's why became the intraocular pressure as a risk factor that cause neurodegenerative changes at the level of the retina, primarily at the level of the photoreceptors, the nerve fiber layer, and the optic nerve. And these results in a late functional disability. In other words, the glaucoma progresses very slowly to the extent that the majority of patients are not aware of glaucoma at the early stages. And they usually, when they are aware of the symptom, it's too late, because about 40 to 60% of the neurodegeneration have already been happened. So the key part of the treating glaucoma is to try to treat the glaucoma at the earliest stage possible before the complete damage happens. How glaucoma is being developed? Well, we know very well that The intraocular pressure is maintained by a fluid inside the eye. We call it aqueous humor. This fluid is produced by area. If you look to the upper right corner picture image, it's a magnified image showing a structure called the ciliary body. The ciliary body has ciliary processes. These are the area where the production of aqueous happened. Then the aqueous travel, if you follow the arrows on the same picture, they release from the ciliary body, they go in front of the lens, they cross to the pupil, over the iris, and then they go through multiple channels, starting from what is called trabecular meshwork, then canal of Schlemm, then collecting canals, then to the venous system. And this aqueous fluid is responsible for the pressure inside the eye. In other words, if there is an increased production of aqueous from the ciliary body, this can result in an increased intraocular pressure. Or, more commonly, there is a normal secretion of aqueous, but the outflow at the trabecular meshwork and the other areas is being obstructed, or actually is reduced. So that's why the majority of treatments for glaucoma are focusing on one of two things. Either you try to reduce the production of aqueous humor through addressing the ciliary body, or try to facilitate the outflow of aqueous from the eye through multiple procedures or treatments that addresses the enhancement of the outflow from the eye. There are two types of glaucoma, what we call open-angle glaucoma or angle closure glaucoma. Luckily enough, the angle closure glaucoma is a very severe condition. It's an emergency, and really this is the one that is associated with a lot of systemic even symptoms in the form of headache, nausea, vomiting, and it's, as I mentioned, it's an emergency. It requires immediate presence in the ER. There are some procedures need to be done in order to reduce immediately the intraocular pressure, and later majority of these patients require surgical intervention. But the more common type is what we call open angle. And when we speak about the angle, it's the angle between the back of the cornea and the iris. The more this angle is narrow, the more patients are liable to increase intraocular pressure and forming glaucoma. So that's the two types are known: primary open angle glaucoma and the angle closure glaucoma. How we manage it, I mentioned the concept is really very simple. Either we try to reduce the production of aqueous or try to facilitate the outflow of aqueous from the eye. And we can do both by either medical, eye drops, for example, or sometimes now we can use eye implants inside the anterior chamber to reduce the intraocular pressure. Or we do laser, and the laser treatment usually focusing towards the trabecular meshwork to create like small holes where it can add to the outflow of aqueous from the eye. Or also MIGS. And the MIGS, this is the area that witnessed a lot of innovation over the last, I would say, 7-8 years, because it is characterized by characterized by simple operations, by very good safety profile for majority of the MIGS. But more importantly, they are divided also about whether we are following the same outflow of the eye from through the trabecular meshwork, which actually should be the best way, or try to create an alternative channels. So in other words, you make bridges from inside the eye to the outside in order that the aqueous can exit the eye. And then we have also major surgical interventions like trabeculectomy. This is the one is known as the gold standard from surgical procedure. And in very severe cases, we have also shunt operations, which really it's for final stages when all the other measures have failed. Next slide. The last point I want to address is the prevalence and the incidence. Glaucoma is a very common disease. And as I mentioned, they call it the thief of sight because majority of the patients don't really know, and they lose vision very gradually, and they don't feel it because it does not affect the visual acuity, it affects visual fields. So majority of patients could hold good visual acuity up to late stages, and only the visual field restriction happen from the sides, goes very gradually so they don't realize it. However, those patients that reach to the irreversible damage or reach to a late stage of visual affections is about 35%. And the best way to treat these patients that have visual acuity is to try to treat them early. Thanks now to a lot of diagnostic modalities like OCT, like many other things, we can diagnose anatomical changes before the functional changes happen. And consequently, we can target the 4.2 million population before they get to the late stage of the visual affection. Because half of this 35 that for patients of glaucoma with vision affection, half of them have an irreversible damage and probably irreversible vision loss. So that's why we need to focus more on the earlier stages before reaching. And unfortunately, also, we don't know which patient will move into those are having the severe visual affection. So that's really where we are trying to play with the glaucoma neuroprotection. That's why you will hear glaucoma neuroprotection. This became one of the major unmet medical need in glaucoma, and many now we're trying, but I think we are having the advantage is going to be the only product with a dual mechanism of action, lowering intraocular pressure, as well as affecting the neurodegenerative component of the eye. Now I will hand over to my colleague, Maisa Attar, to tell you about the BL1107 and how we will address that.
Great. Thanks, Yehia. So where I want to start is talking a little more and building on what Yehia just talked about and why we think BL1107 is the right therapeutic strategy. So glaucoma, as we've heard, is a multifactorial optic neuropathy that's characterized by distinct pathological pathways that all converge on neurodegeneration. So what that means is retinal ganglion cells and their axons sustain injury from various physiological insults. And as we just heard, the risk factor we most commonly recognize is elevated intraocular pressure, which causes mechanical damage. But there are other important disease pathways, as depicted on the right of this slide, for which there is no treatment. And these pathological pathways lead to cellular dysfunction, and ultimately, when left untreated, the cells die. And because the neurons cannot regenerate, the resulting vision loss is permanent, making glaucoma a leading cause of irreversible blindness worldwide. Preclinical and clinical data suggest that alpha-2 adrenergic agonists may help treat injured cells before the damage becomes irreversible. Under normal conditions, activation of alpha-2 adrenergic receptors helps regulate the sympathetic nervous system by reducing release of neurotransmitters such as norepinephrine that's released during stress responses. An alpha-2 agonist drug leverages this natural mechanism and in glaucoma specifically may help suppress these neurodegenerative pathways to support neuronal survival and functioning. So again, while today we treat the risk factor of elevated IOP, glaucoma is ultimately a neurodegenerative disease. And studies have shown that most patients with mild to moderate glaucoma have macular damage and difficulty seeing in low light. So what that means, as Yehia has just said, you want to intervene earlier. So this is the stage where you might have injured cells. So you can still see, but they're not functioning as well. And there is no therapy approved to treat or prevent this vision loss associated with glaucoma. Now, the LOGIC study showed that glaucoma patients treated with brimonidine were less likely to develop visual field loss compared to those patients treated with timolol, even when IOP was decreased to the same extent. Brimonidine is an alpha-2 adrenergic receptor agonist. And alpha-2 adrenergic receptor agonists are thought to preserve and improve vision via two mechanisms. First, neurofunctional enhancement, and second, neuroprotection. Our goal with BL1107, which is a next generation alpha-2 agonist, is to fully realize the potential of these mechanisms. So, BL1107 is a molecule that's designed to treat and target vision-threatening disease. The graphs at the top of the slide show non-clinical pharmacokinetic data that demonstrate when BL1107 is administered as an eye drop, we detect the prodrug and little active drug in the tears. That means the prodrug is available to penetrate deep into ocular tissues, delivering the active drug to the retina, as shown in the second graph, where you see the majority of the drug detected in the retina is the active moiety. Another BL1107 attribute is its receptor selectivity and potency, which is depicted at the bottom of the slide. The lower graphs compare potency to two different alpha adrenergic receptors, alpha-2A and alpha-2B, for BL1107 versus brimonidine. BL1107 is more selective and potent to alpha-2B, while brimonidine is more selective and potent to alpha-2A. And this is important since alpha-2B is the most widely expressed alpha receptor in the retina, where we need the drug to act to improve and protect vision. So it's BL1107's molecular attributes of enhanced retinal penetration combined with selective and potent alpha-2B agonism that should maximize vision benefits. So to that end, Bausch Lomb scientists have continued to generate data to demonstrate how the active moiety of BL1107 protects retinal cells from neurodegeneration. What you're seeing here in this slide are data from an animal model, where rats are exposed to a phototoxic blue light, and we're able to assess the effects of this injury with and without BL1107 active drug. If we focus on the graph in the middle of the slide, we are monitoring structural effects on the retina using spectral domain OCT. The y-axis of the graph measures retina thickness, and the first column with the gray dots depicts normal thickness in a rat's retina when they have no blue light exposure. The next column shows structural damage after blue light exposure, which shows up as a thinning of the retina. In the last column, we see that when we have BL1107 active drug on board, the retina is able to maintain normal thickness, so it protects those neurons in the retina. If we move to the graph on the far right, this depicts a functional measure. Specifically here, we're measuring electrical signaling in the retina, and the y-axis depicts the amplitude or the strength of that signal. The top gray line shows normal signaling when no blue light exposure is experienced. And if you look at the very bottom green line, it depicts the diminished signal after blue light exposure with no active BL1107. And the green line that's just below the normal gray line depicts how signaling is maintained when active BL1107 is on board. So what these data demonstrate is that BL1107 active drug preserves both retinal structure and function following a blue light damage. And so these mechanistic data that I just walked through underlie the positive proof of concept clinical data we've seen in glaucoma patients treated with BL1107. So in addition to lowering IOP, for the first time, we have clinical data that show 15-letter gains, which again is an approvable endpoint. The graphs on the left show that under low light conditions, patients treated with BL1107 show a greater proportion of 15-letter gainers than patients treated with timolol in both the study eye and the treated fellow eye. The top graph depicts the study eye and the lower graphs are the treated fellow eye. And you can see the green bars that represent 2 BL1107 doses on day 1, 4, and 14 you can see that the percentage of patients gaining vision and the gray bars represent vision gains with timolol. And what you can clearly see is more patients are gaining vision with BL1107 treatment. And if we move to the graph to the right, we see that patients treated with BL1107 showed statistically significant improvement in visual field mean deviation on day 14 as compared to timolol. Recall, Yehia said this is one of those early functional measures that you see. And here we're seeing BL1107 again being protective. These unprecedented data led us to run a larger phase 2 study, which is ongoing to demonstrate these visual neuroenhancement effects. The ongoing study is a randomized, double-masked, parallel group study with approximately 159 patients. It's a phase 2 study that's designed to replicate the clinical POC data with refined study elements, such as powering for vision endpoints. We anticipate in the second half of the year to have data from this study. And these data will guide the phase 3 study design. If approved, BL1107 could define an entirely new category of glaucoma therapy. It would be the first with a neuroenhancement mechanism of action aimed at protecting and improving vision while also lowering IOP. And so BL1107 has a robust future. Like we said, the phase 2 study that we're currently running reads out at the end of the second half of this year. And with this powerful pharmacology, we are also exploring indication expansion opportunities for BL1107, such as in retina. And to that end, there's ongoing efforts to develop an intraocular sustained release BL1107 formulation. So the key takeaways are there are no current glaucoma therapies that address the underlying neurodegenerative pathology that threatens vision. And BL1107 has the potential to establish a completely new class of treatment, the first therapy with neuroenhancement mechanism of action in addition to IOP lowering. The clinical proof of concept has already shown BL1107 improves vision in glaucoma patients, and the ongoing phase 2 trial, where we will replicate those findings, is expected to read out in the second half of this year. Thanks. At this point, I want to hand off to my colleague, Kelly Swain.
Thank you, Mesa. As Yehia mentioned at the beginning of this presentation on the anatomy and physiology, I'll dive a little bit more into detail on the MIGS procedures. So MIGS procedures lower intraocular pressure by enhancing the eye's natural aqueous humor outflow pathways, either through the trabecular meshwork, into the suprachoroidal space, or into the subconjunctival space. There are also some approaches that reduce aqueous production via ciliary body treatment, as Yehia mentioned. As we'll discuss further, ELIOS, our laser procedure, works through the trabecular meshwork. MIGS generally are characterized by a favorable safety profile with minimal tissue disruption compared to traditional, more sort of intense glaucoma surgeries, and a meaningful IOP lowering effect that is relatively easy for both patients and physicians. And to that end, I'd like to ask Dr. McCabe how MIGS has changed her treatment of cataract patients who have glaucoma.
Yes, thank you, Kelly. It's been a game changer in how we view cataract surgery, along with the whole picture of a patient who has glaucoma. So now that we have highly safe and efficacious treatments to use at the same time as cataract surgery that also address a patient's risk of going on to having further vision loss with glaucoma, it becomes part of the paradigm of how we intend to treat those patients. We don't just view them as something that we need to do to improve their vision with cataract surgery, but we can also improve their quality of life and their risk profile for progression with glaucoma by addressing their glaucoma with a MIGS procedure.
Thank you. So go to the next slide. ELIOS is well positioned to expand this market. particularly since around 20% of patients who have cataract surgery also have glaucoma, and currently less than half of all cataract surgeons are performing minimally invasive surgery for glaucoma. As mentioned earlier, ELIOS will be our first clinically validated excimer laser in this space and will provide new options for physicians and patients. And again, Dr. McCabe, if you don't mind, as more physicians adopt mixed procedures, how do you think they will decide which procedures will be best for their patients?
Yeah, it's going to be looking at the two most important things, which are efficacy and safety, along with their ability to quickly and effectively adopt the surgical procedure that's needed. So I think these are all areas where ELIOS will help expand the number of surgeons who feel comfortable with the procedure, because the techniques that are needed to be effective are readily available techniques to anterior segment cataract surgeons, and because we don't have to worry so much about positioning in the proper space a device that needs to be left behind. And I know you're going to cover that a little bit more, but that's a key part of the differentiation of this technology.
Perfect. Let's go right to the next slide. We can talk about that. So our excimer laser provides a precise implant-free approach with several features that do distinguish it from other MIGS. One is the ablative laser creates 10 microchannels in a trabecular meshwork to decrease restrictive outflow. Two, the implant-free procedure reduces the risks of poor placement and leaves the trabecular meshwork largely intact to preserve its biomechanics. And as we mentioned previously in the Investor Day in November, this innovative approach offers an opportunity for cataract surgeons who were not previously performing this procedure to do so relatively easily. And then finally, the procedure allows for further interventions for refractive cases because you have, as mentioned before, that anatomy is largely intact. And I guess, Dr. McCabe, which of these features, you mentioned a little briefly, but which of these features do you think resonates most with surgeons?
Yeah, I mean, I've been involved in the MIG space since pretty much it was developed, and I've helped bring different technologies to market and teach a lot of different surgeons how to use different devices and things. And there are a few things that are part of the hurdles that block adoption. And one of those is really, where do I put an implant? Most of the MIGS procedures that were early to the market involve leaving something in the eye. And the problem with that is if you don't put it in the right place or you don't put it in the right way, it's not effective. And beyond that, you can actually have some more long-term effects of leaving it in the wrong place. So doctors don't want to complicate something that they feel very successful with already, which is cataract surgery. And I think, having something that's implant-free, that's readily adoptable in an area that they understand, the trabecular meshwork, and most can identify very readily, I think those kinds of things, that simplicity of the procedure, are really the things that are going to decrease some of the barriers that surgeons currently feel.
Okay. Thank you. And if we go to the next slide, as you've seen in the press release this morning, we're very pleased to announce positive data from our U.S. pivotal study where we've met our primary endpoints, which is very exciting. As you can see on the left, there was sustained IOP reduction by over 20% over the course of 2 years. And on the right, you can see the reduction in medication burden as well, with over 80% of patients that were medication-free. Obviously, we're pleased with these results, and they are consistent with other studies in the EU, where we have 8 years of long-term data showing the durability of effect as well. So this is all very consistent data with what we've seen before, but we're very excited for this well-controlled study to read out positive. And we'll have full results that will be published and released in the coming months. Related to this, Kathy, if you'd like to talk about the data, what's most impressive to you, that'd be great.
Yeah, I mean, there's two kind of things that we look for. Of course, we want rigorous data that's collected in a clinical trial setting and showing that there's meaningful and effective pressure lowering and medication reduction. The thing that matters most to patients is medication reduction, really. That's what changes their lifestyle. That's what changes compliance and therefore efficacy. But the other thing is what happens in a real world setting, and is that durable? Is that something that's going to last longer than the 2 years that we currently think, that you could even get an effective pressure lowering from cataract surgery alone? And what we see is not only is the amount and effectiveness that we see at the 2 year data mirrored by what we see in that 8-year data, but it is durable across the 8 years. So I think this is really exciting data. I think people will really be moved by that. Some of the data that we currently have for other devices really goes out to 5 years. So having 8-year data already is very meaningful.
Great. Go to the next slide. Just the key takeaways I'll highlight again is we really know Elios's position to expand the MIGS market by providing a glaucoma intervention that can be performed with cataract surgery. We also know that fewer than 50% of cataract surgeons are performing MIGS today, and so there's a huge opportunity there. And of course, we have our first clinically validated excimer laser offering a highly precise implant-free approach, now with 2-year data, and we expect U.S. approval in the second half 2026. If we go to the next slide, I think I'll turn it back to Yehia. Yehia, you're on mute.
Thank you. So obviously, just to conclude on these two products, obviously glaucoma still have a lot of unmet medical need that we need to look at the neuroprotective and the neurodegenerative part of the disease. And this is where BL1107 really, hopefully, if the results come as we wish, that could change the way we are treating the glaucoma from a medical perspective. Similarly, on the Elios side, it's going to be the first clinically validated implant-free excimer laser technology, which again addresses a lot of areas, especially with relation to the technology itself, that currently does not exist on the market. And so far, we are on track actually for the launch dates for Elios as well as for the data top line on the second half of this year for the BL1107. So with that, actually, I will give it back to you, Larry, if you want to discuss the questions, if there are questions on both programs, and we'll be very happy to answer with the team here.
Great. Thanks, Yehia, and thank you to the team here for the excellent overview. Again, if anyone has a question they want me to ask on their behalf, please email it to me. I thought I would start with BL1107 and then go into Elios. So starting with BL1107, what have you learned from the proof of concept study that gives you confidence in the phase 2 study, and what are the study design elements that you refined for phase 2?
Yeah, maybe this question I will direct it to Maisa, if you would like to comment on this.
Sure. So our confidence is really based on the clinical findings that demonstrate beyond reduced IOP, BL1107 treated patients showed statistically significant improvement in visual field mean deviation when compared to timolol. And importantly, we also saw those 15-letter gains, which are clinically meaningful. And if this is reproduced in a phase 3 study, it's an endpoint that supports approvability. And so if you recall, under the low light conditions, we saw that patients treated with BL1107, a higher proportion achieved 15-letter gains than those treated with timolol. And so those two findings, with the understanding of how this drug is designed to achieve the findings, as well as mechanistically the way it's supposed to work, gives us a lot of confidence.
That's helpful. Did you want to add, Yehia?
Yeah, I was just going to say also, overall, I think there has been in the past long history, multiple attempts to address this area, but currently there has been a lot of new innovations in diagnosis, as well as new endpoints like the low luminance endpoints. We can detect patients at an earlier stage. So this gives us a bigger opportunity now with the characteristics of the product that Maisa said, plus the data we have from preclinical and clinical to make that this product can work in a better way, or a better population. So we hope that this also translates into our results that we are hoping for all.
And when can we see the phase 2 readout? It sounds like it's top line the second half of the year. I don't know, full presentation maybe at AAO. And how quickly can you move to phase 3?
Again, Maisa, you can answer how quickly we'll move to phase 3. And then obviously for the top line, yes, second half. And hopefully by the academy, we will have some results there, yeah.
Yeah, we obviously recognize that this would be a tremendous opportunity for patients. And so after the data readout, our goal would be to implement the new tools we have at Bausch Lomb to get into phase 3 as soon as possible. So you would see us back in the clinic within a quarter or two of the study readout.
Got it. And what is the regulatory pathway and broad strokes, the timeline you've shared with us in the past?
Do you want me
Go
to
ahead,
say
Maisa.
that? Okay. In terms of the it's a new drug application pathway that we would take through to go through that. Now, having said that, we also recognize that glaucoma is a serious vision-threatening disease, and when a glaucoma patient experiences vision loss, it's irreversible. So we would actually also consider, depending on the data that come out, filing for breakthrough designation. And why that's important is if we're able to do that, we'll be able to have an even faster development pathway. So right now, with a normal pathway, we're anticipating, I believe we've communicated 2031, but our goal, again, depending on the data that we receive, is to accelerate this in every way possible.
Got it. How much, if you have breakthrough, how much time does that save?
Well, the evaluation by the FDA itself could be approximately 6 months saving from the whole evaluation process. It could normally take 1 year, so it could be like 6 months rapid review cycle. And obviously, with the breakthrough technology, you have to do only one study. So this can also save some time, because doing one study versus 2 pivotal could be a big difference in terms of recruitment time and things like that. So we could save some time there as well.
Okay. So broad strokes, maybe potentially saving 2 years in total. Is that?
Yeah, this will be a little bit of a stretch, but around, maybe a year and a half could save that.
Okay. And you showed some impressive data from the proof of concept study. Put that into context for us in terms of how it compares to other glaucoma drops in terms of efficacy and safety. And obviously, you talked about a brimonidine, an alpha A2 agonist. I know it's not an alpha 2B agonist, but how does it compare? That's Alphagan, Combigam, the brand names that are on the market. How would this compare?
Maisa, would you like to comment on this?
So that's a great question. So brimonidine is a drug that's been on the market for a long time, and it's always had hints of neuroprotection, right? But it's been unsuccessful because the clinical effect size is small to actually gain it as an indication, right? And so how BL1107 compares to brimonidine, and for that matter, to any other drug, is that in addition to lowering IOP, which it does, just like any of the other IOP lowering drugs, it actually has, because of its design, it's able to elicit clinically meaningful, statistically significant effects on vision. That is unprecedented. So nothing compares like BL1107, including brimonidine, because BL1107, its design, it's a better design to treat vision-threatening disease than brimonidine, although it does harness the same pharmacology, but more effectively.
Can I add a little bit to that as well? It's just that I want to say how important it is, a 15-letter improvement in vision. And clinically, if you think about that, that's 3 lines. That's like taking somebody who sees their best corrected vision 20/25, which is near perfect in the way we think about vision, and then having them, if they're 20/50, a 3-line worsening, they will be no longer able to pass a driver's test. So hugely impactful on their independence. And if we can instead bring them back to 20/25 vision, an improvement in 15 letters or 3 lines, they then have all that functionality again. So a 3-line improvement is a massive improvement in how it affects their quality of life.
Yeah. And I think Maisa mentioned it during the presentation as well, but I really would like to emphasize, we do believe that our molecule, because of being a prodrug, And the prodrug has a lot of advantages because the normal drugs, when you just pass through the cornea, the majority is being absorbed by the cornea. So the amount reaching to the eye itself usually is much less. But here with the prodrug, the active drug happens inside the eye. So it really gives you a much better targeting and focus inside the eye for the target tissue. And additionally, we said also, I think Maisa mentioned, the potency of this product is really pretty impressive on the retinal cells. At least this is what we saw from the preclinical data on animals. We hope this translates as well into the human, but the preliminary one from the clinical study is still showing that. So that's the difference.
You know, I was surprised to see a 15-line improvement in vision at, by day 14. I think about glaucoma as a slow progressive disease. How do you show an improvement in vision so early, so quickly?
Yeah. You would like to comment on this? This is a great question. So Maisa, would you like to?
It is a great question. So this goes back to this concept of neurofunctional enhancement. If you recall, the reason you lose vision is first the cells experience, because of these neurodegenerative stressors, they become sick, right? They have cellular dysfunction, and if it's left untreated, they go on to die, and then you can't do anything. What our data suggests is we're intervening early enough that we are treating those sick cells to turn down those stress pathways so that they can go back to functioning and seeing again. So essentially that's the neurofunctional enhancement aspect. And if you're doing that chronically, what it will do is it will protect the cells from dying. And so that's the neuroprotection aspect. So that's how we are able to see it so early.
So non-functioning cells are not necessarily dead cells.
Yeah.
They may become dead cells, but if we can bring them back to function, they won't.
Yeah, you think of them as lazy cells. They are unable to perform the function. You want to prevent them from moving from lazy to dead cells. And you're trying to catch them at the lazy stage where you just try to activate them to be just the normal cells again. That's really what we are trying in a very simple terms.
Okay. That's super helpful. So we've got about 17 minutes left. Maybe we can transition to Elios, and if we have time, we'll come back to BL 1107. Elios is the more near-term opportunity. Obviously, I'm primarily a device analyst. This is probably a little bit closer to home for me. Maybe let's start with where you see Elios being positioned in the current treatment landscape. I don't know if that's a question for Dr. McCabe or whoever.
I think Dr. McCabe would be the best to answer this one.
Yeah, and I'm happy to answer it because, I see Elios as the go-to interventional glaucoma device that surgeons are going to use in conjunction with cataract surgery. And we really have a mindset that's changed a lot over the last years in that if we have the opportunity to get a patient to have IOP control with minimal medicine intervention, that decreases their burden, cost, and compliance-wise, and increases the likelihood of efficacy and protection from further visual loss in the future. Because Elios has excimer laser, which is very precise, and we really feel that. We use excimer laser for LASIK. We understand how precise it is. We know it's a non-thermal laser. And it creates more openings in the trabecular meshwork than any other thing that we have, while preserving normal tissue for further intervention. And the technique needed to do that is very accessible. I think it will be the go-to device that we usually think about. Let's have one thing in our back pocket that we really feel good about, that we know we're going to be able to effectively do, that we're going to have the least side effects, the least risk of complications. And I think this will be the device that people look to do that.
And there are other laser technologies out in the market today for full glaucoma, SLT, for example. How is this different?
I'm happy to take that, and if you want to jump into, DSLT is different. It's a different market. It's for earlier intervention. It's an in-office procedure and has more of a variable response. I think one of the things we're most excited about Elios is the short learning curve that Kathy's spoken about, as well as even for those who are not as experienced with MIGS procedures, and the favorable reimbursement profile of this product. So we're excited for that. And as mentioned before, the precision with the excimer laser itself.
Got it. And you showed the 2-year, Kelly, you showed 2-year data today for the first time. I haven't had a chance to look and see how that compares to the other MIGS. There's a lot out there. I mean, I think people are probably most interested in iStent, and since that's the biggest. But if you want to put that into context with goniotomy, canaloplasty, et cetera, that would be helpful.
Absolutely. So happy to give a little bit of overview and reinforce some of those points. And then Yehia, please jump in, and Dr. McCabe as well. So as mentioned, we shared the positive 24-month data on Elios today. The safety and effectiveness of this data is similar to that of other MIGS devices, but without leaving an implant behind. So I think that's one of the really exciting things about this data. We met our primary effectiveness endpoints, demonstrating both statistically significant and clinically meaningful IOP reduction. The unmedicated diurnal IOP was reduced by 20% or greater in 76% of the patients, so the majority of patients saw that effect. Patients experienced an average of 7.4 millimeters mercury decrease in unmedicated diurnal IOP. And the average number of medications went from 1.5 at screening down to 0.3 at 23 months, so a very large reduction there. No intraoperative complications seen during the procedure, and a similar postoperative adverse event profile from cataract surgery alone. So I think all in all, the data is consistent with what we've seen outside of the U.S., but even more reinforced with the well-controlled study that was run. And Dr. McCabe, if you have anything to add or...
Yeah, the only thing I would also add is that the 2-year data that we see in that 8-year published real-world data is very, very similar. And one of the things we saw early on with stent-based MIGS interventions was it looked like sometimes the efficacy was not quite as good. And that's because a lot of those stents you can actually put in the eye, they'll stay in the eye, but they're not necessarily in the tissue they're supposed to be. So I think one of the things that's going to be a real advantage to this is it is a more direct application right to the trabecular meshwork, where you know that you've done the treatment where it needs to be, and you have 10 rather than 1, 2, or 3 fenestrations that you've created, but without destroying all of the anatomy, which is what a large goniotomy does.
Dr. McCabe, what's the learning curve here? You talked about it earlier. You said efficacy and safety are really important, but so is ease of use. And I'm paraphrasing what you said. How does that the ease of use and the learning curve stack up to other MIGS procedures?
Yeah, and I've had the opportunity to do a lot of training on lots of different MIGS devices, but I've observed a lot of people who have used Elios as the first time that they've used it. And it could be very experienced glaucoma surgeons, experienced in the angle. Some of those doctors were doctors who had started off with stents and then just gave up on it because they didn't feel comfortable with it. And then other doctors that I observed were new users in the angle of MIGS procedures. And all of them found that it was very accessible, very similar to other things that we need to do as far as how you move the instruments in the eye. It's just simply a probe. We use probes for lots of things. And you just need to identify the trabecular meshwork, which is easily identifiable. So every single one of the doctors I observed, and myself, as I learned this procedure as well, found it to be, very, very straightforward. So I think the learning curve is going to be very easy.
That's helpful. And maybe another one for Kelly. How confident are you in obtaining regulatory approval and launching Elios in the second half of this year?
We remain confident and look forward to the launch. Yes, second half.
Okay. You talked about reimbursement. Somebody mentioned reimbursement. This is a space that's had a lot of volatility, to put it mildly. Forgive me, on the pivotal trial, was it a single arm trial or was it an RCT? And what's the how do you expect the reimbursement to shake out compared to other MIGS?
I think it has a specific reimbursement codes as a procedure. So that's actually one of the biggest things that we always look at. And Elios has already one. I think the trial, to your answer is question, it was actually a trial that had a single arm. So it was a single arm of doing combined cataract and glaucoma surgery. And I think we have a very good reimbursement structure so far present. I wish that we had somebody from the commercial to speak more to this. But generally speaking, I think the reimbursement is based on even the trial that we have. So we think we have a very good chance to achieve the desired reimbursement for the product.
So we have a strong reimbursement team that understands procedural codes and understands procedures such as this. We have confidence in the procedural code that we will use at launch. And so we know that this will be a competitive product within the space and certainly competitive with all the other MIGS devices that exist today.
Okay. I mean, I guess is there anyone, do you have, with this code that you have, do you have any advantages on the reimbursement side compared to other technology that we should be aware of? Do you think it'll be competitive, Yehia, to other like iStent, or do you have advantages on the physician fee?
I know it's I cannot comment to the competitive part, but I know it's going to be very good in terms of the reimbursement part for Elios.
Yes.
That's
an R&D call, so we'll follow up with considerations on the.
Yes, I think that's the best way. Yeah.
But we all know on this call how important the reimbursement is in this space.
I would just say that the reimbursement we anticipate will be able to support the enthusiasm that we expect surgeons to have for the technology. And sometimes it's a barrier. We don't think reimbursement will be a barrier. We'll just support that enthusiasm.
That's fair. That's good to hear. Is there an opportunity here as a standalone procedure for Elios, or do you envision this mostly as a done in combo cataract?
Yeah, it's like for now it's a combo cataract. That's what was studied in.
Is there a plan or hope to study it as a standalone procedure going forward?
So right now we don't have plans for that, but we are looking into that as a future development.
Got it. And before I go back to BL 1107, just the $175 million peak sales. Would anybody care to kind of characterize how to think about the ramp? Are there any analogs you would point to? Is this going to be fast, slow? Any comment?
Yeah, Larry, maybe I'll answer that one. I'll jump in here. So we do anticipate the approval in the back half of this year. I really think you're going to see the impact in our P&L in 27 and ramp nicely in 27 into 28. I think given the learning curve that Dr. McCabe described, I think it will ramp more quickly than other MIGS, but it will still really be a 27 P&L impact for us in a positive way. Yeah.
Got it. Thank you. That's helpful. And then going back to BL 1107, we didn't talk about the sustained release formulation. There's a lot of excitement with the success, iDose is having. We have Durista out there. Talk about the status of the sustained release formulation and how it might compare to the incumbents.
Yeah, I think I will hand over also to Maisa to give an overview of this. But before I hand over to Maisa, I just would like to mention if the study results by the second half of the year comes as we expect, this will open the door for multiple other indications. So it's important for us also to start to think about other formulations other than the eye drop. And this is where we also started to focus on drug delivery system, long acting. And I think I'll just hand over to Maisa to give you also a perspective on this program that we are doing.
So, sure. So in terms of our work on the sustained release, it's the data so far in our hands are very promising and support entering development at the end of this coming year. Oh, that's right, at the end of this year, 2026. And how we compare, I think it's inherent to the molecule that we're formulating. So if you recall, we also have a partnership with Ripple that really has best-in-class approach for long-acting injectables. And because this would be the first molecule that's being formulated to lower IOP as well as protect cells in the retina, nothing can compare with it. And the doors it could open, it could be a long-acting glaucoma treatment. It could be a long-acting retina, like geographic atrophy treatment. So there's so many things we could do with a formulation that was long-acting. And so, again, I don't think there's anything like it right now. And it really comes back to what we see in this first clinical study that's going to read out later on this year to confirm mechanistically it's doing what we have seen it do so far. And then we are very excited at the opportunities that will open for us.
And then maybe lastly, could someone talk about the IP and the patents you have around BL 1107, please, and how long they go out?
Yeah, I think for the BL 1107, we actually have numerous pending families around the world, with several have been issued. We also have a lot of new applications for composition of matter, pharmaceutical composition of claims, methods of treatments, new mechanisms, potential other indications. So we have a very wide patent portfolio for BL 1107. It goes all the way up to 2046. And actually, we are actually, some of them are pending, but some of them have been already granted. So we are moving in a very good direction with this one in terms of patent. I think Brent mentioned also at the GP Health Morgan that one of the areas that we're focusing on across all our new pipeline is patents. And this is an area we have seen a dramatic increase, unprecedented number of new patents we have applied for all our products during the last year, and this year we continue to do the same. So for BL 1107 in particular, we are very confident, and it goes up all the way up to 2046.
That's helpful. So 3 minutes left. I wanted Yehia to give you kind of an opportunity to add anything that we didn't discuss that you want to highlight or to summarize. If I do the math here, these two assets, it's about $1 billion peak, $800 million. I think you have for BL 1107, 175 for Elio. So I'm rounding up a little bit, but obviously, big opportunity here for Bausch Lomb. Thank you again for the opportunity to host this. But Yehia, I'll give you the last couple of minutes here.
Thank you very much. We really appreciate the opportunity, Larry. And again, we are really very excited about our pipeline overall. I think we are really trying to target areas that have been known to be a high unmet medical need. There is not a lot of approved treatments currently. If we speak about the neuroprotection piece, we think that this is an area, obviously, a lot of companies looking, but this will be the first product that have dual mechanism of action to address the IOP as well as the neuroprotection piece of glaucoma. And similarly, also, I think for the cataract surgeons that are looking for a quick, fast MIGS procedure without leaving an implant behind, with a very high precision, a very rapid process, and also a very effective for a very long period of time, the Elios is a great approach to that. And I think we are actually trying to find out even more to increase our pipeline with such things that are unique, differentiated, and addressing big areas of unmet medical need that we are still not addressing. So that's really where we are focusing with our pipeline. And we will have more sessions like this for other products, I hope, in the near future as well.
Sounds good. Well, with that, we'll end it. Appreciate everybody's time today, and hope everybody on the line found it useful. So with that, we'll end the call and see everybody soon. Thank you.
Thank you.
Thank you.